Good morning, my dear students. I am Dr. M. Venkat, Assistant Professor in Zoology. Welcome to my Aniruddha tutorials. I teach zoology for the NEET entrance examination, All India Medical Entrance Examination. I hope you will learn more about zoology for the All India Medical Entrance Examination. Thanks for listening to my class and follow my class. Thank you all. My dear students, again welcome to my Aniruddha tutorials. I am Dr. M. Venkat. Today I am going to deal with the locomotion and movement. Locomotion and movement Basically, what is the meaning of locomotion? What is the necessity of the locomotion? The organisms shows locomotion What is the necessity of the locomotion? For the search of the food material, maybe for mating, to obtain the suitable climatic conditions, optimum climatic conditions, in search of a optimum breeding place, to get the breeding ground, and to avoid the enemies, to protect from the enemies, Well, this is the necessity of the locomotion. In search of the food materials, maybe for the mating purpose, to get a optimum climatic conditions, in search of a suitable breeding ground, and from the protection from the enemies. So before I discuss about the locomotion, what is the meaning of the movement? Movement is a sort of the movement of the body. Locomotion is movement of the organism from the place to place is called as the locomotion. Therefore, in organisms, please see a simple form of movement. The simple form of movement is the streaming of the protoplasm. In case of unicellular organisms like amoeba, in which the streaming of the protoplasm is seen. It is nothing but the rotation of the protoplasm. It is the simple form of movement. It is a very simple form of the movement. It is the streaming of the protoplasm, like in amoeba. Amoeba is a unicellular organism, belongs to phylum Protozoa. In amoeba, streaming of the protoplasm is seen. In organisms, there are different types of the locomotory organelle. Generally, There are cilia, a type of the locomotory structures, and one more, flagella, also the pseudopodia. Pseudopodia, cilia, and the flagella. Pseudopodia help in the amoeboid locomotion. These are involved in the amoeboid locomotion. Amoeboid locomotion is performed by the pseudopodia. Flagella are involved in the flagellar locomotion. Ciliary locomotion is with the cilia. This is called as the ciliary locomotion. Ciliary locomotion occurs by the cilia. Flagellar locomotion occurs by the flagella, and the amoeboid locomotion performed by the pseudopodia. It is very important. Please see, the amoeboid locomotion is not only involved with the pseudopodia. There are some other cytoskeletal structures. There are cytoskeletal structures called as microfilaments. Microfilaments are the cytoskeletal structure, which are the cell skeletal structures are microfilaments, which are proteinoid, and these microfilaments are also involved in the amoeboid locomotion. I repeat again, amoeboid locomotion is generally performed by the pseudopodia. In addition to the pseudopodia, there are some other cytoskeletal structures are called as the microfilaments are also involved. And the flagellar locomotion occurs by the flagella, ciliary locomotion occurs by the cilia. For example, some protozoans like the paramecium, in which ciliary locomotion occurs. Flagellar locomotion occurs. Example, one euglena, flagellar locomotion. Automatically amoeba performs the pseudopodial locomotion or the amoeboid locomotion. And these are some locomotory organelle in the lower organisms. But here, the simple form of movement is the streaming of the protoplasm is the simple form of the locomotion. In the human beings, we see some locomotor structures inside the body. Inside the body, we have some locomotor structures. In human beings, we have the amoeboid locomotion. And also ciliary locomotion, flagellar locomotion, amoeboid, ciliary, and flagellar locomotions are also present inside the human body. Some cells, some parts of the body are exhibiting this amoeboid, ciliary, and the flagellar locomotion. For example, in the human body, we have the leukocytes. Leukocytes are nothing but the WBC, white blood cells. They exhibit the movement. White blood cells, they move from place to place. Generally, they have the amoeboid locomotion. And also macrophages, which are present in the connective tissue, which are the phagocytic cells, macrophages. They are also exhibiting the amoeboid locomotion. Inside the human body, the ciliary locomotion is present. Cilia are present in the wall of trachea. Ciliary epithelium, it is lined by the columnar ciliated epithelium. is lining the trachea. What is the purpose of the cilia inside the trachea? Because cilia beats not in this direction, not towards the lungs, away from the lungs, because to prevent the purpose of the cilia in the trachea is to prevent the entry of dust particles as well as the microorganisms into the lungs, entry of the dust particles and the microorganisms into the lungs, and this is the purpose of the cilia in the trachea. What is the purpose of the cilia inside the trachea? Yeah, trachea is lined by the columnar ciliated epithelium. The purpose of the cilia is to prevent the entry of the dust particles as well as the microorganisms. And coming to the cilia, also present in the inside the fallopian tubes in the female reproductive system. Fallopian tubes also lined by the simple columnar ciliated epithelium. Simple columnar ciliated epithelium lines the fallopian tube. What is the purpose of the cilia inside the fallopian tube? What is the purpose of cilia inside the fallopian tube? For the purpose of cilia in the fallopian tube is for the movement of ova. Towards the uterine cavity, and also the ventricles of the brain and central canal of the spinal cord, these two are lined by the same simple columnar ciliated epithelium. These two are also lined by the simple columnar ciliated epithelium, but that simple columnar ciliated epithelium that is lining the ventricles of the brain and the central canal of the spinal cord is called as ependyma. Ependyma is nothing but the simple columnar ciliated epithelium, but in the central nervous system. Ventricles of the brain, cavities of the brain, the paraceles, the first two ventricles in the cerebral hemispheres. And the diocele is present in the third ventricle, that is present in the diencephalon. Fourth ventricle, the bilocele present in the medulla oblongata. Therefore, the ventricles of the brain, nothing but the cavities of the brain. In human body, there are four ventricles. First two ventricles are present in the cerebral hemispheres. They are called as the paraceles or the lateral ventricles. Third ventricle is present in the diencephalon, called as the diocele. Fourth ventricle present in the medulla oblongata, can be called as the myelocele. And all these ventricles of the brain are lined by the ependyma, and the central canal of the spinal cord also lined by the ependyma. What is the purpose of the ependyma? In the nervous tissue, for the movement of cerebrospinal fluid movement occurs inside the ventricles of the brain and the central canal of the brain with the help of the ependyma. Remember, ependyma is nothing but the simple columnar ciliated epithelium, and this is all about the cilia in some parts of the human body, lining the trachea, lining the fallopian tube, lining the ventricles of the brain, and also the central canal of the spinal cord. And coming to the last one, flagellar locomotion, I write here. Flagellar locomotion is seen in the flagellar motion is exhibited by cells. Spermatozoa of male in the motion absent in the female because flagella present in the because tail wriggling motion occurs. And coming amoeba. WBC macro inside the trach fallopian tube. The ventricles are central spinal cord. Vaginal locomotion exists spermatozoa. And coming important. So the reproductive exhibiting fallopian tubes. Inside the fallopian tube there is a ciliary locomotion. I write here. Ciliary locomotion. Ciliary locomotion in fallopian tube. Because for the movement of the ovum, fallopian tubes are present in female reproductive system. But what type of movement is present in male reproductive tract? The male reproductive is vasa, the male tracts, and urethra, etc. In these are the male reproduction epithelium. There are no cilia. But in the male reproductive tracts, locomotion is present. Generally, nothing muscular movement. Muscular movement occurs. Male ciliary locomotion occurs because fallopian tube is ciliated epithelium. Epididyme, vasa differentia, urethra, urinogenital tracts are never lined by the cilia, but that movement is nothing but the muscular movement. It is very important. And coming to one more. The locomotor structures need not be different from affecting the other kind of locomotor structures. Locomotor structures need not be different from affecting the other kind of locomotor structures. Because, for example, if you say this statement as assertion. Assertion says that locomotor structures need not be different from affecting the other kind of locomotor structures. The reason is, for example, the cilia in paramecium of paramecium help in not only locomotion and also for the food capture. See, cilia are okay locomotor organelle, but also helping for the food capture. And the tentacles in hydra, help in locomotion as well as the food capture. Tentacles are also performing the two different functions. Cilia are performing the two different functions, and even our limbs help in changing the body posture and also for the body movements. See, the locomotory structure, one structure is performing the two different functions. That is why locomotor structures need not be different from affecting the other kind of the locomotor structures. Cilia are performing the two functions and our limbs are performing the two functions. That is why assertion and reason both are right, and it's a correct explanation. Lastly, all locomotions are movements, or all movements are locomotions. A statement. Statement 1. All locomotions are movements. It is a correct statement. And one more statement, for example, statement 2. All movements are locomotions. All movements are locomotions. It is a false statement. I repeat again. All locomotions are movements, but all movements are not locomotions. That is why all movements are locomotions. It's a false statement. And coming to the human body. In the human beings, musculoskeletal system, musculoskeletal systems and neural system are coordinated for the perfect body movements. I repeat again. Musculoskeletal systems and the neural systems are coordinated together for the perfect body movements. In our body, even though some parts contain the ciliary movement, amoeboid movement, and also in the males spermatozoa exhibiting the flagellar movement, but most of the body locomotion is under control of the muscle. Muscular movement is the major movement. Muscular locomotion is the major. You know pretty well some characters of the muscle. Muscle is the only one tissue. Muscle is the only one tissue exhibiting a property called as the contraction, and that property is the contractility. Even though human body contains different types of the tissue, but only one tissue undergoing the contraction, exhibiting the contraction is the muscle. This property is for only muscle. There are some characters exhibited by both muscle and the nerve. One is the excitability, and also conductivity. These two characters are shown by both muscle and nerve. Both muscle and the nerve are exhibiting these two properties. One, excitability and conductivity. This property is shown by only muscle. Contractility is by only muscle. There are some other properties. One in the extensibility and elasticity. Extensibility and elasticity, there are other characters in the muscle. The character which is exhibited by only muscle is the contraction, which is shown by the both muscle and the nerve are excitability and the conductivity. Even though these two are shown by the other tissue, but also present in the muscle, extensibility and the elasticity. And one more important property, muscle is generally mesodermal in origin. Muscle is generally mesodermal in origin because there are some exceptions, but not all muscles are mesodermal. In the origin of the muscle, some exceptions. One, iris muscles of the eye and ciliary body of the eye. These two muscles are derived from ectoderm. They are derived from ectoderm. So, I am reading these characteristic features of the muscle. Study the characters of the muscle and choose the one without any exception for muscle. Muscle contractility, yes. This character is present in only muscle. Muscle and nerve exhibiting the excitability and the conductivity. But all muscles are mesodermal. Wrong statement. Most of the muscles are mesodermal. Correct statement. There are some exceptions in the origin of the muscle. Iris muscles of the eye and the ciliary body of the eye are not mesodermal. They are ectodermal in origin. This is all about the characteristic features of the muscle. In our body, human body, 40 to 50% of body weight is the weight of muscle. In the total weight of the body, 40 to 50% of the body weight is the muscle weight. And now I want to categorize the muscles. Muscles are classified in the different criterion. Yes. Basis of the classification. Basis of the classification. Depending upon the location, depending upon on the basis of the location, on the basis of the appearance, on the basis of appearance, and on the basis of the regulation. These are three criteria for the classification of the muscle. Depending upon the location, depending upon the appearance, and depending upon the regulation. On location, muscles are three types. Depending upon location, muscles are three types. One are skeletal muscles. Second, visceral muscles. Third, the cardiac muscles. Skeletal muscles, visceral muscles, and cardiac muscles. These are the three types of the muscle, but on the basis of location. Depending upon the appearance, muscles are two types. Depending upon the appearance, muscles are two types. You see, this is a striped muscle, yes. Skeletal muscle is a striped muscle depending upon the appearance. Cardiac muscle also a striped muscle. Visceral muscles are unstriped muscles. Depending upon appearance, muscles are two types, striped and unstriped. Depending upon the regulation, there are voluntary muscles. Skeletal muscle is a voluntary muscle. Smooth muscle or the visceral muscle is involuntary muscle. Involuntary muscle. Cardiac muscle also the involuntary muscle. So, depending upon the regulation, muscles are two types. One are voluntary and involuntary. Depending upon the appearance, striped and unstriped. On location, skeletal, visceral and the cardiac muscle. And coming to the structural aspect of the skeletal muscle, I am going to deal with the structure of a skeletal muscle as follows. Structure of a skeletal muscle. For example, the biceps muscle or the triceps muscle, skeletal muscle. This is the skeletal muscle. The structure is as follows. This is the skeletal muscle. But skeletal muscle is externally covered by a sheath, a connective tissue sheath around the skeletal muscle. And that sheath is epimysium. Epimysium. This is the outermost connective tissue sheath. Okay, I write this is the hierarchy from lower to higher. Okay, this is muscle, skeletal muscle. If you take the sectional view of the skeletal muscle, I take the PS. It is as follows. Again, this is the muscle. Okay, this is the epimysium. Epimysium, and this is the muscle. Inside the muscle, bundles. This is a bundle. They are called as the muscle bundles. And these muscles are also called as muscle bundles, are also called as the fascicles. So I write, these are the fasciculi, are the muscle bundles. But around each fascicle, there is again a connective tissue sheath. And that connective tissue sheath around that fascicle is called as the perimysium. So, epimysium, muscle, perimysium, and muscle bundles are fasciculi. What is present inside the fasciculi? Before I discuss that, the various fascicles are connected by. This is a fascicle, I write here. A fascicle. The fasciculi are internally bound with the connective tissue. The connective tissue which is binding the fasciculi called as the fascia. Fascia. Fascia is a connective tissue but what kind of connective tissue? It is a collagenous connective tissue. A collagenous connective tissue between the two regular fascicles is called the fascia. Then what is present inside the fascicle? I take this is the fascicle, one muscle bundle. Yes, this is the muscle bundle of the fasciculus. I told you a tissue layer or around that every fasciculus is called as the perimysium. Actually, this is the perimysium over the fascicle muscle bundle. What is present inside the muscle bundle or the fasciculus? Each fasciculi contain the muscle fibers. These are muscle fibers. So I write muscle fibers. Muscle fibers. Each fascicle contains the muscle fiber, muscle fiber, muscle fiber. But around the muscle fiber, again they connective tissue sheath is present. This is the connective tissue sheath, connective tissue sheath, and the connective tissue sheath around the muscle fiber. And the sheath over the muscle fiber is the endomysium. Endomysium around the muscle fiber. See, I take one muscle fiber. This is a muscle fiber. I write muscle fiber is wrapped by a sheath, and this sheath is the endomysium. Sheath around the muscle fiber is the endomysium. Then what is present inside a muscle fiber? Each muscle fiber contains parallel structures are called as the myofibrils. Fiber contain the myofibrils. Myofibrils are present in the muscle fiber. See, this is the muscle fiber. Fiber contains a parallel structure. This is a myofibril. Myofibril. There are different myofibrils. What is present inside a myofibril? Myofibril contain the myofilaments. Myofibril contain the myofilaments. And this is the myofibril, internally contain the myofibril. Then I want to discuss about the structure of a myofibril. This is the myofibril. Yes, structure of a myofibril. Structure of a myofibril. This is myofibril. Myofibrils are the structures which are parallel to each other but have the bands, light band, dark band, light band, dark band, light band, dark band, light band, dark band, light band, dark band. They are alternatively arranged. So this is called as the light band, thin band, light band. Light band also called as the thin band. Thin band also called as the I band. What is the other name of the I band? I band is called as the isotropic band. Isotropic band or the I band. And this alternative structure, this is called as the A band, dark band. Dark band is called as the thick band. Thick band is the isotropic band or A band. So this is A band. I repeat again. This is the I band and I band and A band are alternative. And isotropic properties are the live index property. They look like homogeneous is isotropic. If it is not homogeneous, it is heterogeneous. This is anisotropic. Active index property of the light. So I band, light band, A band, dark band, anisotropic band. I band, A band are alternatively arranged. This is fibril. And now I want detailed structure myofibril. See, if I want to take this is the I band and A band and another I band. I take one I band and another I band. This is band and A band, one more I band. This is I isotropic band or band, light band. This is A band or anisotropic band or the dark band. Again, this is the I band. I band, A band and I band. So this myofibril is lined by the plasma lemma. Yes, it is called as the plasma lemma, sarcolemma. Plasma lemma. What is present at the center of the I band? I band is equally bisected by a line. I band is equally separated by one line, and that line is called as the Z line. Z line is also called as the Dobbe's line. Dobbe's line also called as the Krause's membrane. Krause's membrane. Z line, Dobbe's line, Krause's membrane. But this Z line is a elastic line. Z line is one elastic line. Again, at the center of the I band, this is another Z line. Z line and the Z line. The distance between the two regular Z lines, distance between the two regular Z lines is called as the sarcomere. Sarcomere is the distance between the two regular Z lines. Two regular Z lines is the sarcomere. Two regular Z lines is the sarcomere. What is sarcomere? Sarcomere is nothing but the functional unit of the muscle contraction. Right here, sarcomere is the functional unit of muscle contraction. Very much important. Sarcomere is the functional unit of the muscle contraction. If you are given a statement, both structural and functional unit of the muscle contraction, wrong. It is only a functional unit of the muscle contraction, sarcomere, the distance between the two regular Z lines or two alternative Z lines. And coming to the A band. At the center of the A band, there is a zone. And this zone is called as the H zone, Hansen's zone or H zone. At the center of the H zone, there is a line. And that line is M line. As I told you, Z line is elastic. Z line is elastic line. But M line is thin and fibrous membrane. Very much important. It is a thin fibrous membrane. M line. It's all about the structure of a myofibril. Then each myofibril contains the many myofilaments, and now I will discuss about the fibril inner side contain the myofilaments. What are myofilaments? Myofilaments are two types. Myofilaments. One are the thin filaments. Second, thick filaments. Thin filaments and the thick filaments are two myofilaments. Where are they present? See this. Thin filament in the entire I band Thin filaments are present in the entire I band and also a part of the A band, but not in the H zone. When the muscle is under rest, thin filaments are present in the entire muscle in the H zone. Then up to the boundary of H zone, edge of H zone. This is a thin filament. See that filaments are present in the entire I band, also a part of the A band, but not the H zone, H zone. Thin filaments are never present in muscle is under resting condition. I think you understood that. Hello? And one more. This is the thin entire I band, thin band, extending up to the edge of the H zone. A band also consists of the thin filament, and this is another thing, the location of the thin filament. Repeat again, the thin filament. Filaments are present in the entire I band and A band, but not the H zone. And coming to the thick filament, are present only in the thick filament. This is the thick filament, only A band, present in only A band. But thin filaments are present in entire I band and a part of A band, excluding the H zone. Remember, when the muscle is under resting condition, thin filaments are present in the entire I band and also a part of the A band, but excluding the H zone. Thick filaments are present in the entire A band only. These are two types of the myofilaments, one thin filament and the thick filament. And now I have to discuss about the total structure of a thin filament and the structure of a thick filament. Structure of a thin filament. Structure of a thin filament. I told you that thin filament is present in the entire I band and also a part of the A band, but excluding H zone. One thin filament contains one thin filament contain two number of the F actins, filamentous actins, and two tropomyosin, two F actins and two tropomyosin, and many, these are in number, not one or two, etc. They are many in number, but these are many complex proteins. I repeat, complex protein called as NEET. These are in the thin filament. One thin filament, F actins, two tropomyosin, and a complex at this regular interval complex protein. I show you a monomer. This is a globular protein called the G actin. Globular G actin. G actin is the monomer. G monomer of as many G's are forming one filamentous actin. G actin many agree form a totally one actin. One can the actins. These F actins. Hence, the monomer, who is the polymer? F-actin is the polymer. F-actin is the polymer, G-actin is the monomer in the thin filament. These are two F-actins. Over. Two tropomyosin. See this. This is myosin and this is another. If we see the length of the tropomyosin, these are myosins. On the tropomyosin, I repeat, these are complex proteins are called as the troponims. The troponims are present on the tropomyosin. Okay, this is a troponin and this is a troponin. Troponims are present on the tropomyosin. Right here. Troponims are present on tropomyosin. Very much important. Why troponin is a complex protein? Because each troponin has three subunits. This is a subunit of the troponin, subunit and another subunit. There are three subunits of the troponin. Hence it is a complex protein. If you see one troponin, for example, if I say this is the tropomyosin, on the troponin, the subunits of the troponin, if the troponin is reacting with the calcium ions, subunit of the troponin reacts with the calcium ion is called as troponin C for calcium. The subunit which is interacting with the tropomyosin reacts with the tropomyosin is called as troponin T, tropomyosin. And this is the subunit of the troponin which is one inhibitor is called as troponin I. It is inhibitor subunit. It inhibits the binding of the myosin during the muscle contraction. It inhibits the binding of the myosin head. And it is called as the inhibitor because that is site of the actin. So the one thin filament contains myosin on the complex protein are present. It is a complex. Each one troponin has the three subunits. Reacts with the tropomyosin. Troponin C reacts with the calcium ion. In I, it is a molecule. This is the again in the thin filament, the G-actin polymer is the F-actin. In this thin filament, both troponin and tropomyosins are called as the both troponin tropomyosin troponin and tropomyosin are called as regulatory proteins. Called as the regulatory proteins. It is the statement of assertion. Assertion says that both the troponin and tropomyosins are the regulatory proteins because why? Because the reason is troponin and tropomyosin molecules can mask or close or unmask, open. They can close or open. They can unmask or close or unmask. They can close or open. They can close or open. They can unmask or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask or open. They can close or open. They can unmask Troponic tropomyosin can mask and unmask the active sites of actin. They can mask, unmask the active sites of the actin. If you see, there is an active site of the actin that can be closed, can be opened. Both the tropin and tropomycins are masking the active site, opening the active site. That is why they are called the proteins. So, my dear students, I think you understood my lesson. Thanks for listening to me.